Thanks all. It's a great pleasure to be here, and I will do my best to keep you awake post-lunch, which is always the good energy dip in the middle of the day, but maybe I'll say enough provocative things that it'll keep your blood pressure up well enough. So at any rate, the title of my talk was the working title of my book, Science Friction. The point being that the things I discovered in thinking about reproducibility and rigor in science doesn't say that science has stopped at all, scientific progress, but that these things are slowing down science, and if we could ease the friction, we could speed up science. I'll talk to you today about my explorations around those issues and what we can all do about it. So I'm going to start with a story of Tom Murphy, whom I met reporting a story for NPR. In 2014, when I was asked to turn my attention back to biomedical research, I thought, I'll take a temperature and see what's been going on in biomedicine since I had really stopped paying attention. I covered it on and off through my career, but mostly in the 80s and 90s, and was doing environmental reporting mostly in the interim. So I thought, well, see what's going on in science. And one of the stories I came across was the story of Tom Murphy, who had been diagnosed with ALS, Lou Gehrig's disease. He knew that he basically had one shot at a clinical trial because people with ALS generally live only 4 or 5 years, and their health steadily declines. And once your health has declined sufficiently, nobody who's running a clinical trial wants you in it. They want somebody who's relatively healthy so they can more clearly see any trends that they have in their drug. So he had to choose from which of these clinical trials he would choose, and he got on a trial, and it failed. In fact, it didn't really matter which trial he chose; they all failed. There's been very little progress in ALS, and one reason, as I came across him partly because I was looking down, upstream of this clinical trial, and it turns out that some folks have been looking at the mouse models that underlie many of the ALS studies, and this is a paper from Nature that was put together by folks at the ALS Therapy Development Institute in Cambridge, Massachusetts, and they asked, these are all mouse studies that had been done on the potential drugs treatments for ALS, and these were all turned out to be failures, and they said, well, what's going on? And they decided, let's take a closer look at the mouse studies. And one thing they discovered was many of these mouse studies were done with a half a dozen mice, very small number of mice. People were not doing some of the basic things like making sure that if you have litter mates of mice, that you put them in different groups in your experiment as opposed to lumping them all together, and doing some of those basic hygiene, laboratory hygiene sorts of things. And so they said, we're going to do this right. We're going to use many dozen mice per sample. Costs a lot, well over $100,000 per test, which is something that not everybody else had the luxury of doing. And we'll do these right, and we'll see what our results are. And this is what they came up with. This is change in survival up here. And what they showed is basically, when you do the mouse studies right, essentially none of these were promising drugs at all, with the possible exception of riluzole, which actually is on the market already, has some minimal benefit for ALS. But basically, these guys said, why do we spend all this time doing all this preclinical research if we're not going to do it right? Because all we're doing is leading ourselves in the wrong direction. So this was an important wake-up call, I think, for the world of ALS. It was also an important wake-up call for NIH, as it turns out. This is Shai Silberberg, who had been on a committee that was funding one of these clinical trials. I think the NIH spent $40 million on this trial, and it was a bust. And he sat in on one of these prep sessions for a clinical trial, and he noticed that everyone started talking about clinical trial design, making sure the design's right, which is of course critical, but he realized nobody was asking the first question, which is the preclinical research adequate? Have we done our legwork to make sure that we have done the preclinical studies are done with enough mice and all these other things? And he came away with the conclusion that no, in fact, no one's paying attention to that. You get to this point in the NIH process, and people are thinking ahead. They aren't thinking back and asking those important questions. So he raised this concern at his institute, and his boss, Story Landis at the time, recognized, yeah, this is a big issue for us. And she started proselytizing about the need for really careful thought about rigor and reproducibility at the NIH. And she really, he and then she ultimately got this really on the agenda for the NIH. And I think very much to the benefit, I think, of the long-term health of science. And it's just started out with this Israeli-born scientist who was a electrochemist or something like this, who wasn't quite as disciplined, but he was a biophysicist who did very precise work in laboratory, and was a little surprised to see about how animal studies were not treated with as much care as he certainly had expected. So, of course, this didn't start with all that. This is a paper that was raising the alarms in 2005. This is John Unitas, who's now at Stanford, and his essay, Why Most Published Research Findings Are False, really looks at some of these issues from a statistical point of view and points out how many papers in the literature are really very badly underpowered, and we draw too much from them. And I should say that this talk is talking mostly about preclinical research, because I think that he found in this paper, which is really a thought experiment more than anything else, and the general sense is that in clinical research, people thought through these issues a lot better, and they've made a lot of improvements since the 1990s. And so the issues I'm talking about really are in preclinical research. But this was a wake-up call. This is his most famous paper, gets a lot of attention. Again, people don't necessarily notice it's a thought experiment, not data-driven, but it still stirs the pot quite a bit. And this other paper also stirred the pot a lot. When it came out in 2012, Glenn Begley, at the time, was head of cancer research at Amgen. And he'd been there 10 years, and he said, okay, I'm ready to move on and do something else. But before I leave, I want to go back and look at some of these papers that have crossed his desk over the years. They prospect in the scientific literature. Drug companies increasingly rely on university researchers to come up with exciting, interesting ideas to pursue as drugs. And he noticed over the years of doing that so few of the ideas, these exciting ideas that get reported in the scientific literature, actually worked by when he tried them in his laboratory, and they were potentially really exciting drug leads, but they essentially, so many of them were dead ends. So he said, I'm going to go back and I'm going to look at about 56 of these studies and give them another go, run them through again, see if we can get them to work in our lab. If we can't, let's get in contact with the original scientists and ask them to redo them, see if we can come into their labs and watch them get it done and so on. And he had some cooperation along those lines. And in the end, he was only able to reproduce 6 of these 53 or 56 papers. I've lost the exact number, but about a little better than 10% was his success rate from this. And this helped also raise the issue about reproducibility in an area we really care about, sort of the early clinical or preclinical work leading toward treatments for cancer. And I will say one thing about this paper: it is also not reproducible, because he won't say what the papers were that he was examining. He promised people anonymity in exchange for including them in this survey. But there was a similar study also done about the same time by the drug company Bayer in Germany, and they came up with a very similar number. They got about 25% reproducibility out of the papers they looked at. But the point is, this was an awakening that this is a significant issue, and that we need to grapple with it, because it's the friction in science. We need to reduce this friction. And so in the course of reporting my book, I talked to a bunch of people about this. I'm not just out on a limb caterwauling here. This is Janet Woodcock, who heads drug approval for the US Food and Drug Administration, and she was complaining to me about, in particular, academic research. She says, drug company scientists know much more what the rules are. They know how careful they need to be to get through the entire FDA process, and not her words, mine, that basically what they lack sometimes in creativity, they make up for in precision, because it really matters. It's the incentives are for them to do all of these studies carefully, less so academics. And she was saying when she gets a drug proposal that's been backed by academic research, it really drives her crazy, because she thinks most of the time they're wrong. And her quote to me was, it's like 9 out of 10 airplanes we designed fell out of the sky, or 9 out of 10 bridges we built failed to stand up. We need rigorous science we can rely on, she said. And her colleague across Maryland a little ways, Francis Collins, who heads the NIH, also has embraced this as an important issue. And he and Larry Tabak, his chief deputy, wrote in one of the Nature journals some pretty strong words, I thought. We all know that in the long run, science is self-correcting, but he said, in the shorter term, the checks and balances that once ensured scientific fidelity have been hobbled. Those are strong words to the guy who's running the NIH. So as I say, much to his credit, instead of trying to sweep this under the rug, he's really embraced this as an issue and tried to make rigor and reproducibility something that scientists or NIH grantees think more carefully about. And there's certainly been progress along those lines. This is not just relegated to the world of biomedicine, of course. This is a paper that got a lot of attention a couple years ago of psychology experiments. And these guys decided to try to reproduce about 100 psychology papers. Kind of not really a great, sort of a grab sample. They just asked themselves, what paper do you want to reproduce? And so they sort of came up with 100 papers that they were interested in, but were not systemically chosen. And they found, or systematically chosen, and they found that of those 100, they got about a third of them to reproduce correctly. About a third, actually the statistics weren't strong enough to say whether they were right or wrong. And the other third, actually some of them, quite a number of them were pointing in the other direction, and actually from the original conclusions, finding the exact opposite effect. So this is an issue, this is Brian Nosek's work largely at the University of Virginia, and he's done a lot also to think about how to grapple with these issues as well. So Nature decided to ask the question of 1,500 researchers, is there a reproducibility crisis? And more than half said, yes, there's a significant crisis. 38 said, yes, there's a slight crisis. I'm not sure exactly what a slight crisis is, but a number of people seem to like that idea. And then about 10% said, I don't know, I don't care, I'm washing my hair, call me back tomorrow, whatever. So, but the point is that this is certainly perceived in the world of science as a crisis. I actually think this is not the right way to frame this. I actually don't think it's a crisis. It's an awakening, because I think these issues have really been with us for a long time. They may be getting a little bit worse, or maybe considerably worse, because of some of the funding pressures that in particular biomedicine has suffered in the last decade or so. But I think this is an endemic issue, and I think there's now an awareness about how big an issue this is, and that's all for the good, because once you are aware of an issue, you can then take steps to correct it. So. This is the cover of my paperback, Rigor Mortis, with the publisher's preferred title as opposed to mine. But basically the tale is in that I'm telling here is largely in that book. So what are the causes? Well, bad ingredients, designs of experiments that are susceptible to bias, statistical errors, and bad incentives. And I'll talk about each of these in turn. You may recognize this famous picture of Henrietta Lacks, who in 1951 had cervical cancer and was treated at the Johns Hopkins Hospital. They took a sample of her cells and were able to propagate them in the laboratory, and her cells, the HeLa cells, became the first immortal cell line in use in biology, and have been used phenomenally successfully around the country and so on, and around the world, as a matter of fact. There's a problem with these cells, though, which is they're kind of the kudzu of the cell world, because they grow so much faster and more aggressively than practically any other cell that you find in a laboratory. That is, other scientists developed other cell lines. Very often they accidentally contaminated with HeLa cells, and so what they thought they were studying one cell line turned out to be the HeLa cells, and this became a huge problem in the awareness of which was coming out in the 60s and 70s, actually, and was also largely ignored by the scientific community that didn't want to hear that they were having a cell line contamination problem. But it really the tools at the time were harder to use, because you had to look in the microscope and study the shape of the chromosomes to really understand whether your cell line was HeLa or something else. And obviously the genetic tools have increased tremendously, and it's actually now trivial to do a genetic test. You can send it off to a commercial lab, and for $130 or something, they'll tell you whether your cell line is what you think it is. But HeLa cells ended up just being this incredible contamination problem. And just a year ago, these guys in the Netherlands decided to go back through the scientific literature and said how much of the HeLa cell line misidentification contaminates the scientific literature. And they went back to see there are entire cell lines that are just like, for example, Chang liver is used in a bunch of scientific experiments. You'd think it was a liver cell. It actually turns out it's HeLa, and that the whole cell line is HeLa cells, but people have studied it for many years as Chang liver, and there are a bunch of these. There are well over 100 cell lines that are just simply named something but are actually HeLa. And so they said, well, how many of these papers are in the literature? And they identified 33,000 articles of the easiest ones to identify, and those in terms were referenced by 500,000 other papers. I don't think that they were able to grapple with one of the major cell lines that was a contamination. It's called KB cells, which also turn out to be HeLa cells. But KB is also, if you search for that in the scientific literature, you end up with kilobyte, right? So you end up really not being able to identify cell lines very well. But the point is, they just found this enormous amount of what they call contamination in the literature of these papers that purport to be about one condition and are actually based on studies of these cervical cancer cells. There maybe are some circumstances where it is not so important whether you're studying a cervical cancer cell or a glioblastoma, but if you think you're studying glioblastoma and you're not, you're in trouble here, and there's a lot of papers like that. So, and there's a group called the International Cell Line Authentication Committee, which tabulates these misidentified cell lines. There's Chang liver, Hep2, KB. You can see a bunch of them. There are more than 400 that have been identified. About 100, or a little more than 100 are HeLa, but there's tons of other cell lines out there that are misidentified. They're even the wrong species often, that people think they're studying human cells and they're actually studying completely different species. So this is actually a problem that's easy to resolve, and the NIH has actually taken steps to make sure that to tell scientists, we expect you to check your cell lines and make sure that you're studying the cell line you think you are, whether that means sending samples off to commercial labs or the core laboratory at your university. Do something to validate your cell lines, and that's an expectation in grants. I have no data to know how often scientists are actually meeting that expectation, but that is now one of the clear and easy things that NIH said, this is one thing we can do to deal with reproducibility issues in the world of cell biology. So I mentioned statistics. Here's a statistics example. It's called Harking, hypothesizing after the results are known. At first glance, sometimes people say, that doesn't seem like that big a deal. You get your data, you rummage around in it, and you see something interesting, and voila, there's your result. But this is actually a pretty dangerous practice because it completely blurs the line between exploratory research and confirmatory research. And if you are testing a hypothesis, that should be a confirmatory study. And this is, rummaging around in your data is a good way to explore, but don't then mistakenly say, I have data to support my hypothesis. And the illustration I have is this cartoon that ran the paper not so very long ago about the young archer who's shooting an arrow at the fence, and she happily hits a bullseye. This is the consequence of harking. If you do it wrong, you can always paint your bullseye around wherever the arrow lands, and you have to be very careful about doing that. There are lots of p-hacking. There are a bunch of other statistical things that I'm happy to talk about more, but there's a whole list of these things that fall into this problem of statistics. And I was talking to Keith Yamamoto, who is a muckety-muck at UC San Francisco, who's been in the field for a long time, and he said, a lot of my peers and I went into biology because we really didn't want to do math. So we thought, biology, if you can't see the results in your data, think of a better experiment. And that was the dark ages of biology, and he now recognizes, as we all do, that it's become an intensely mathematically driven field. You really need to understand statistics and how to analyze stuff, and the world is still catching up to folks like that. And I talk to a lot of people who really, even young scientists who admit, I wish I had a better statistics class. I took the class, we went through it, and then that seemed to check the box, and we went forward, and appreciating really how much more time and effort not just statistics, but also thinking about experimental design. That these are issues that that people look back and they say, I wish I'd had a little bit more of that in my training. And it's starting to happen in a few places, but I also think that it's it's once it's also disruptive, because if you're going to have more classes in experimental design, you're going to either have more time in grad school or less time to do other things, and it's a complicated issue, but it's all baked into what we're seeing here. I'm not portraying scientists as a bunch of who the heck cares what's going on? I'm getting my grants and I'm moving forward. This is it's built into the whole system of science, and this is one place where we see this arises, and it's another problem that we can recognize and fix, and that's all for the good. So in terms of incentives, this is actually a curve that I came across when I started doing my reporting, when I came back to my beat in 2014, which is, if you look at this graph, you can see that between fiscal year 1998 and 2003, the NIH budget actually doubled in real dollar terms, or in spending power terms. It was a phenomenal time for biomedical research. And people went on hiring sprees and building sprees. The amount of lab space that is a consequence of this, the amount of biomedical lab space in this country increased by 50%. Largely on the basis of this huge surge in funding. And then in 2003, Congress said, okay, we've fixed the health problem. We don't have to give them any more money. In terms of real dollars, they flatlined it, but of course, there's inflation, and so the spending power steadily eroded. So over the next decade, after doubling in a decade, over the next decade, it decreased by 20% in spending power. And you have all of these new people in science, all of these new laboratories that have been built and so on, and it made an already very competitive world hyper-competitive. And that has some very negative consequences for the way science gets conducted. Again, not deliberate, malicious, or whatever. It just puts the pressure on people to publish these amazing, fantastic, unbelievable results. And in fact, these guys wrote a paper about this a couple years ago. They were looking at positive and negative words in abstracts between 1974 and 2014 to see whether people felt more compelled to pump up their research findings as scientific competition increased. And they found, yes, indeed, they found words like robust, novel, innovative, and unprecedented increased in relative frequency up to 15,000%. Which, of course, was, I think, their own little joke of that sort of hyperbolic way of expressing this. But the point is that this is a problem that has driven scientists to pump up their work. The press offices at universities pump it up. Journalists are certainly not blameless either in trying to make things seem bigger than life. And the whole effect is that you have more and more emphasis on sparkle and dazzle, and rigor seems to matter less and less. The rewards are for getting these papers in. I've talked to so many people in biomedical research labs, postdocs in particular, who say, if I don't get a paper in Science, Nature, or Cell, I may not have a career after my postdoc. And so can you imagine the pressure involved in finding something that's so dazzling that you can get it in one of those single-word journals? And it's tough. It's a very bad incentive system, and we got to break that if we want to get people to step back and say, I want to do the best science I can, but we can't say, well, you have to do that at the expense of your career. That's a non-starter. So what are the consequences of these problems? One of them is, this is one of my favorite graphs, it's called Eroom's law, which is Moore's law spelled backwards. You may be familiar with Moore's law, where semiconductors get faster and faster and cheaper in sort of a logarithmic scale. Well, this is a graph that makes the opposite argument about research and development efficiency in drug development. And starting in the 1950s, it gets more and more expensive to get less and less output in terms of how many drugs it takes per $1 billion of research expended. Jack Scannell published this a few years ago. And I think that I actually am curious to see what happened past 2010, because I think there have been some more successful drugs on the market since 2010. Certainly, we haven't reversed this trend, but it may not look quite as grim in this as it does. But the point is, and this is not necessarily driven by reproducibility issues. In fact, they're probably just a fairly minor part of this. But the point is, this is the environment where we're working, and you can't, and if you already have this kind of pressure on research progress, what you need to say is, where are the levers? What are the things we can do to help reverse this trend? And one thing is you can reduce the friction, and you can try to make science more effective and get more bang for your buck in your preclinical research in order to help make sure that your drugs that are coming out the pipeline are actually going to work. I mean, I would say something like 9 out of 10 drugs that enter the drug pipeline actually never make it out. And you could change those odds, I think, if you based it on better preclinical research. So that's one consequence. Another is what you read in the newspapers. This is a paper by Estelle Dumas-Mallet at the University of Bordeaux, and she looked at 200, and her colleagues looked at 200 English language newspapers to say, okay, how well have these newspapers captured the correct science? And it wasn't that the science writers were misreporting the results. We were actually reporting the results correctly. The question is, did the results hold up over time? And she did a study and found a whole bunch of studies that have been done over the years. And she said, okay, how many of these have had meta-analyses done? And she found about 150 that had meta-analyses done. And she said, okay, so when we have some better results from meta-analysis, what do you find out? And you find out that of these 156 primary studies, only about half actually turned out to be correct in the meta-analyses. And if you take the flashiest studies, the most exciting ones, the first of their kind, only about a third of those turned out to stand the test of time. So as a journalist, I read this sort of disheartened and said, maybe at the end of it, when I just do a story based on a single study in the literature, which I try to avoid doing, but we all do it sometimes, sort of thinking maybe at the end of it, I say, well, about half the time, studies like this turn out to be right, and about half the time they turn out to be wrong. So are you feeling lucky today? Get my audience to think about the fact that these findings are provisional, right? As we all expect science to be. So what are the solutions? I've already talked about validating ingredients. I think that's an easy and important step, getting the cell lines right. I think there's a whole other world of trying to validate antibodies, which are used very widely in biomedical research labs. There's a huge amount of work to be done to validate them, and some of the big companies are starting to chip away at that. But I think it's also incumbent on scientists in the labs not to expect that the antibody really works the way you think it does, but to run some good experiments to controls and so on to make sure that the antibody is doing what you think it's doing. Transparency, I'll say more about that. Verifying results, I think that's a very important step. And also changing these bad incentives. And I've alluded to that a little bit as I've talked about some of the cultural issues, some of the pressures that are confronting scientists, and what we can do about that. So this is a famous paper, cover from Nature in 2001, when they published basically the first draft of the human genome. Amazing accomplishment. And of course, they didn't publish all 3 billion base pairs in this issue, but they decided, let's pick about a dozen or so exciting results that we found and highlight them in this story, in the study, in the paper that they wrote. And one of the ones they chose to highlight was an observation that they had come up with that basically a discovery of several dozen genes that appear to have gone straight from bacteria into the human genome. A very surprising result. We understand how viruses could take genes and inject them directly into the human genome, but how a bacterium could actually get in its genes sort of tucked into the human genome, not through evolution, but recent inputs, was really quite a mystery to them, and exciting, a very exciting finding for them. And the answer was, well, it actually didn't happen. This is Steve Salzberg and company who said, we've looked at those genes, there are about 40 of them, and guess what? It was an analytical problem. Now, I'm telling this not to sort of shake my finger at the genome scientists, but to say the reason this was resolved, because this was only a few months later, this was June of 2001, the reason this was resolved so amazingly rapidly was the human genome data were all put in the public database, right? Anyone could go to GeneBank and look at it and figure it out. And this is the story of transparency in action. They had a conclusion, they put their data up there for other people to look at, and very rapidly other scientists could say, thanks for sharing your data, we've looked at it, we have a completely different conclusion. And I talked to Francis Collins about the paper in Nature, and he said, yeah, well, we were wrong, but fortunately no humans were harmed by that error, except maybe a few bruised egos. So transparency is something that a lot of people are pressing for as a way of not necessarily avoiding errors, although sometimes if you know that someone's going to be scrutinizing your data, you might take a little bit more care about how you analyze it to begin with. But also knowing that science is 2 steps forward, 1 step back, and this accelerates that process. The more sharing you can have, the faster you get through those issues. And I think it's a good, healthy thing for science to embrace. Again, it conflicts with some of the cultural drives to be competitive and to make sure that you're not giving your competition an advantage over or insights into what you're up to and so on. So there are competing interests, but transparency I think is an important part of the answer to how to deal with these issues. And Brian Nosek, whom I mentioned earlier, is big on thinking about how to improve the transparency and how to make that happen a lot better. And one thing he has is this thing called the Open Science Framework, which he's trying to convince people, if you can start at the beginning, put your data in, you can have it sort of a closed ecosystem for yourself and your collaborators, and then at some point, when you're ready, when you publish, you just push the okay, everyone can look at it button, and it becomes a very easy way to share. I'm not sure this is gaining a lot of traction because it's asking for scientists to do a lot of work up front. He says, well, they normally do it at the end anyway, so why not just sort of change their workflow? But it's hard to change habits. It's hard to convince people that it's worth that initial investment in their time to end up with a system that really benefits all of us, including those of us, everyone in this room, who helps fund this research, right? We are, NIH budget is approaching $40 billion. We have a lot at stake in making sure that they're spending our money wisely. I wanted to touch on Barbara Slusher. One story she told me, which is germane to this meeting, was about what was going on around the year 2000 in the 1990s. She told me a story about some drug companies very excited about developing new painkillers, actually. They got very excited about something called NK1 antagonists, and drug companies poured a lot of money into it. The basic science seemed to be pretty good. The mouse models all seemed to suggest that it worked, and so the drug companies went in whole hog, and basically it was a bust for painkillers. I think there are other places where this molecule turns out to still be of interest in medicine, but as a painkiller, it totally didn't work. And Barbara Slusher's point was that we were relying too heavily on these mouse models of pain and having a sense that if it worked in the mouse, it's got to work in human beings. And the field was wrong about that. And she said that was a very kind of a humbling experience for her because this was one of the areas she had been working in. But she realized, we need to think more carefully about how this happens. And one thing that it certainly happened was the drug companies started saying, whoa, to academic scientists, we're not so sure that we're going to just take your latest idea and run with it. We want to hear more from academics about how sure they are that their results actually have clinical relevance and are significant and at least are reproducible. And so Barbara Slusher, who's at Hopkins, started a program there that has now been replicated across the country where she says if somebody at Hopkins comes up with an idea that is potentially a drug target or whatever, something interesting to pharmaceutical companies, before they ship it out to the drug companies or sell it off or whatever, she says, bring it to my operation here. We're going to validate these results here at Hopkins, and we're going to put it through its paces before we take that next step. Yes, it takes a little bit more time, but the result is that we are more confident in the results going forward. And when I talked to her, this is several years ago, I talked to her, I said, well, how successful has it been? She said, well, about half of the stuff that comes out of Hopkins that comes through my lab actually works. It's like, oh, so about half doesn't, even at Johns Hopkins, which at the time was the #1 recipient of NIH funding dollars. So it's like, I'm glad she's doing that. And it was also sort of a resonance that even the best scientists in the world are not always getting this right, and we need to be aware. But I think this idea of taking the extra time and taking the care to validate results I think that's really healthy for science. People maybe think it's slowing it down, it's not helping my career that much, but I think that there are well over 1 million papers published in scientific literature every year. I don't know about you, but I don't read all of them. In fact, I don't read very many of them. And in fact, I would be happier if there were fewer of them, because I think most of them are out there really to satisfy career needs. And I think we need to think very carefully about different ways to reward people so that their career is not based on how many papers they've published and where they've published it, but some other measure of how good their science is. And I think I would like to, as a funder of this research myself, as a taxpayer, I would like to see fewer papers, but I'd like to see them of higher quality and really make sure that what's out there is more likely to be true. I think that helps us as a society move forward better. And I think her effort, this shows where some of those centers are, is also I think is germane along those lines. Finally, I want to talk about some of the cultural things I've talked about. Those are big, complicated issues to deal with, but I wanted to highlight John Unitas, I mentioned earlier, and his colleague, Steve Goodman, who are co-directors of this thing called METRICS, the Meta Research Innovation Center at Stanford. And their idea was, let's study research, meta research. Our research will be to study research and how research is conducted. And at the core of that is trying to understand these reproducibility issues. And so they have an effort going to really understand where these problems arise and where the pressure points are and how to make changes in the culture of science in order to make science more productive and more frictionless. And I end on this note because I think they were both veterans of the 1990s, where a lot of these problems were plaguing clinical research. A lot of clinical trials were done poorly. The results were hard to interpret. They were very contradictory. They were underpowered. They had a lot of these problems. And people would change their endpoints once they realized, oh, I didn't get the answer I expected. I'll say I was actually testing a different hypothesis. And they point to clinicaltrials.gov as one of the many tools that was put to use in clinical trials to say, okay, tell us up front what you're studying, what your primary hypothesis is, and really help think through whether you're doing good science or not. You put it up for all the world to see. If you change your endpoints, it'll show up as a change in this. And I think that has been one of the important aspects of clinical research that has really helped improve the quality of clinical research. By far not the only thing, but one idea that Goodman and others have had is like, could we take this example, this idea, and apply it to preclinical research? Maybe if we get people thinking on a little bit more rigorous standards of how to get things done, how to make sure that your hypothesis is well formulated, that it doesn't change after you've seen your data, and so on. I think these are ideas sort of learning from the past, from a different world, and saying, how can we apply that to preclinical research? I think all of these things are happening. I think there's a good movement in this direction, but there's also a long ways to go. And I think that people don't necessarily appreciate hearing about the problems in their field, but I think I think it is genuinely. I've had many very hopeful and productive conversations with people about recognizing that these are happening and thinking about ways to improve this, because we all want the same thing in the end. Nobody wants to do bad science. Nobody wants to waste their time or taxpayers' money or anything else. So if we're all thinking a little bit more carefully about these issues, I think everybody wins. So thank you very much. I'm happy to take questions. Stunned silence. Wow. All right. Well, thank you so much for your attention. I'll be. Oh, there's a question. Right.
So those journals
could you repeat the question?
Yes, I said, has there been any push towards making sure that whatever experiments gets accepted into a journal is actually rigorous, because you have scientists sitting on those journals who can look through the experiments, making sure that the method and everything else is actually up to par before accepting those. So is there any push towards making sure that that process is a little bit, actually stands up to time?
There is. The journals themselves, for example, the Nature journals ask you to fill out a checklist and ask you some basic questions like, did you get your cells validated, and some of these basic questions. The trick is that they may publish your paper anyway if it's interesting enough, even if you haven't followed through on the and answered correctly all those questions. But they are hoping that over time people will say, well, next time I write a paper and want to put it for Nature, I know I'm going to have to answer these questions differently. So I think that's providing a little bit of pressure. And I think just there are also guidelines like that have existed for a long time, but people don't necessarily pay a lot of attention to them. There are guidelines about, well, how to conduct a appropriately powered animal study, for example, and those sorts of things are increasingly being considered, I think, when journals publish papers. But it's also true that it's a big change, it's a big cultural change, and it's also, I mean, honestly, if you're a scientist and you have so many pressures on your time and so little money to work with, you can't you sort of. I've talked to many scientists who say, I can't do every single little thing I need. I just can't. It just would make my research grind to a halt. So I do the best I can, and I hope it's good enough. And I think that reviewers recognize that also. So they don't always necessarily say, well, you didn't do this perfectly, so we're not going to run the paper. So it's, I think it's movement in the right direction, but I don't think that. Certainly these new guidelines, these new NIH requirements, and these new, journal expectations are nudging people in the right direction, but they're not rushing in the right direction. Yes?
So I have a question particularly about preclinical modeling. I'll hear your thoughts on two points. One is, to do the studies in mice or other organisms or other animals that you're talking about takes a lot of money.
Right.
And the NIH budgets haven't really reflected that. And if you'd comment on that. And the second comment I was going to ask, or comment I was going to make about the really prohibition now on using primate organisms for model systems, and how that plays into some of the the reproducibilities we translate to human studies.
Right. Animal models are expensive. The ALS TDI experiments I think they were like $130,000 per experiment, which is a huge amount of money. I mean, if you're a university researcher, that's probably out of reach of your grant unless you have an extraordinary grant to do for a single careful experiment. So it's a substantial and difficult issue. But just saying, well, it's a pilot study, I only use 6 animals, but it's okay because I'll call it a pilot study and hope that it's representative of, if I'd actually done the experiment, with scientific rigor, that this is a facsimile of the result I would have gotten. That's not a good result either, and a good way to go either. I think you've put your finger on a very important issue, which is that it again gets back to my argument that I'd rather have scientists do maybe a little bit less, but in substantially more depth. And so maybe instead of running 10 experiments, if you do, have how many projects you have going, think about that. I know it's a difficult thing because it clashes with the academic reward system, and that needs to be changed, I think. So if you're really going to get people to do fewer but more rigorous experiments. In terms of, animal. The other thing about animal studies, and including certainly the primate studies, is there's also the sense that from the standpoint of animal welfare that we want to use as few animals as possible out of compassion for the animals and so on. But that conflicts sometimes with using enough animals actually to get a scientifically meaningful result. And I think that's a conflict that's ongoing. I think people are aware of it. I don't think there's not a clear resolution to that, but I think that's an important issue also to think about when thinking more broadly about these issues. Yes?
Understand that the clinical trial model of specifying everything up front is clear benefits for some of the issues you raised, but over the centuries, science has really taken advantage of serendipity, and how do you maintain that approach without killing the unexpected finding?
Right. I think actually a simple answer to that, which is just be clear about saying this is an exploratory study, not a confirmatory study. I think the weight that you put on those results isn't like, hey, I found a result. The weight is, hey, I found something interesting, and now I'm going to go do a confirmatory study to see if I'm right. And I think that's much of anything, it's a way of honestly portraying where you got to the answer you got and saying, look, I didn't expect to see this in the data. This is interesting, and I want to, now find out doing, if you're going to be applying p-values and all these other things that are the standard hypothesis testing kinds of statistical packages you're using, you want to make sure you're actually testing a hypothesis, not just rummaging through your data. So it's not. I agree that you definitely don't want to snuff out serendipity at all, and you don't even necessarily want to stop doing these kinds of studies. I think you just want to be very careful about how you portray them once you have done them.
You've spoken a bit about p-values, over-reliance on p-values and study design, and obviously testing, doing confirmatory analysis versus exploratory analysis. Can you speak a little bit more about some of the pitfalls of quantitative research that you've seen in your interviews or in your research?
Yeah, well, I think the other area that I think got off to a sort of a rocky start were some of these huge genomic dives, sort of splashing through genomic data looking for patterns, and obviously looking for this schizophrenia gene was the gene, which of course didn't exist, but a lot of people thought they were on the right track. There are a lot of these sort of, if I look through enough data, I'm going to find some interesting pattern without fully appreciating the statistical tests you need to make sure that you're not just fooling yourself and finding some random pattern. I was talking to a guy named Josh LeBaire, who's been very involved in some of this stuff, and he's an editor of a journal who says, if somebody gives me a paper that's a correlation between a gene pattern and a disease, I'll say, I will not publish this unless you actually can give me some mechanistic reason. Identify gene, make me understand why this actually might be a real correlation or a real. Yeah, and not just a correlation. And so I think and John Unitas talked about this to me as well, about the fact that in his view, a lot of the early data in this area was like, he put a number on it, was like 99% of it was just plain wrong. And he thinks this is actually an area where the field has really turned around and completely understood that they have to think about these old problems differently, and he's actually now pretty confident in the studies that are coming out of that world. But if you go back, and certainly people do, into the older literature and expect that those studies to be meaningful and on target, buyer beware. Yes.
Is there any significant amount of funding being put toward replicating existing results?
Is there funding being put toward replicating results? Not enough. I think this happens depending upon the kind of result, but this tends to happen more informally in laboratories, because if you're a new postdoc, you're being, there's some exciting result, and your PI says, let's pursue that idea, but before we do, why don't you replicate the results just to make sure we're on the right track? And this is, I think, very often, if you're in the lab and if you're asking a kind of problem that you can be attacked that way, that does tend to be the way that a lot of people first start out trying to replicate results. One of the problems is that very often those attempts fail, and they don't publish the fact that they failed. So there may be 6 different labs that are trying to chase after an idea that turns out not to work, and they haven't compared notes, and they don't realize we're all wasting our time because no one has taken the time to publish the negative results and said, well, we tried to replicate this and we couldn't. That's beginning to change a little bit. There are journals like the PLOS journals that say, we're perfectly happy to publish the so-called negative results, but a lot of scientists are really not happy about spending the time to write up those papers. But on the other hand, they also recognize that they get the short end of the stick when they spend, it could be 6 months or a year. I talked to a postdoc at a lab who said, I spent 6 months chasing after this result, and I finally realized it wasn't a real result, and it was like, now I got to start all over again thinking about what's my research project going to be in this lab. So yeah, that's the NIH should spend more money, I think, on those things. But if you're talking about competitive grants, these panels are unlikely to say, oh, yeah, I'm going to fund that study even though we think we already know the answer, or somebody else thinks they know the answer. So that's another systemic problem that needs to be addressed for sure.
So recently there was a publication coming out of China that a researcher was able to edit the genes of an embryo, and claiming that these the babies born would be able to, or would be resistant to the HIV virus. I know back in the early 2000s, late 90s, there was another claim that adenovirus was able to cure squamous cell carcinoma just by injecting the virus, also from China. Increasingly, we're seeing more and more publications coming from China and India and other developing countries. And one of the problems many journals are facing is that it's hard to know what institutions these are. There's a lot of pressures in these same universities to publish, particularly in large, highly competitive fields. So I wonder if you could comment on some of these challenges that we face outside of the United States, particularly in places like China, and how that's being addressed.
Yeah, that's an important question. I was talking, in the course of also writing my book, I was talking to somebody who was a journal editor who noted that if Chinese scientists at the time, if they could get a paper published in Science, Nature, or Cell, they would get a bonus worth their entire year's salary. And then often they would sell co-authorships too as a result. And so this was, you think the cultural issues in U.S. science are rocky. There are, I think there are obviously the expectations, the mores in China are very different, and it's a great concern. Honestly, I think that we need to. It remains to be seen whether this CRISPR experiment is actually a real experiment. The results are real. It was released primarily as a series of YouTube videos and then a talk at a scientific conference. It has not been published, so we don't really know about the veracity of those claims. But yeah, I think we need to be vigilant about these about this. I mean, this is the thing, it's plausible, right? CRISPR's not that hard to use, and it's completely plausible that somebody in China would say, our lab doesn't particularly care about these ethical issues, we'll go ahead and do it. And we're not going to try to verify that experiment in this country by replicating it, that's for sure. But yeah, I think that we just need to be aware that the standards are different and the motivations are different, and to the extent that we can be aware of what those are, we can be appropriately cautious about those results. I mean, obviously good science does come out of China, and we don't want to just say, well, just ignore anything that's published in China, but we also have to just keep our antennae up and be aware that it's a different culture, it's a different approach to ethics fundamentally, and we just need to be mindful of that. All right. Well, thank you all very much.